Hmm?
Yes, sir.
Today, do you have any question from the previous class? That of course it is already more than 1 week, I think, time that the from the that morphology of flower and this description of flowering plant. No, one plant belongs to Solanaceae. I taught no that Daturas, Solanum, tuberosum. Mm, do you have any question from there about the drawings, those floral diagram, floral formula from there? Do you have any doubt, any question? Do you have nothing? You don't have anything, no? In the plant morphology that do you remember that in the plant morphology we had something? Okay, if you don't have anything, then today I will do this next that the cell biology portion starting, and here mainly we will uh, we will learn about this cell structure, cell division. Out of this say cell division, that one today we will learn. uh, we will learn cell division. All right, this is very important. And in the reduced syllabus, this cell division, meiosis and mitosis is there. All right. So this we know that the growth and development, is it? Growth and development is the characteristic feature of all living organism. Uh, Along with, of course, the all living organism has several important character. In one of them is the growth, and the other one that is the reproduction. Is it that by which the organism or the species they increase their number and maintain themselves on this planet? Is it by reproduction? And by reproduction that they produce the similar organism. Reproduction, they produce the similar organism. And in this process, organisms are there are unicellular organism, multicellular organism. Multicellular organism, they increase their size by increasing the number of cell, and for that the new cells are to be produced, and this new cell production of the new cell takes place. Production of new cell takes place by cell division. Is it? So, and this is also this producing the new cell. There is also reproduction of the cell, means cell reproduction. I think we know this the cell theory. Do you remember cell theory? In the cell theory, that two very basic and important things are there. That one is cells are the structural and functional unit of all living organism. Is it that all organisms are made of cell or its product. So not or and its product, cell products. This is because all the organism they are that made of cells. So at the cellular level, it is the structural basis and the functional basis. So structural and functional unit. Okay. And the second point was is that the all cell or new cell, okay, new cells arise from pre-existing cells. That when new cells, how they arise from the cell which is already existed, and that happen by the division of that cell from the pre-existing cell by division or by cell division. Okay. So new cell arise from the pre-existing one by the cell division. So this cell division is the very much basis. This for unicellular organisms are either unicellular or multicellular. So unicellular organisms, this is cell division is the basis of multiplication. That is, they multiply, they increase their number, basis of multiplication. But in case of multicellular organism, it is that basis of the growth. Basis of growth, because multicellular organisms are made of many cells, and those cells, cell number increases in an organism due to this cell division. All right, and all the organism start their life from single cell. Say those which are unicellular, their life is that is they are single cell, but the multicellular organisms also begins their life from a single cell, and this single cell they divide and increase the number, and multicellular huge organisms are produced. So this cell division, okay, there are two types of mainly two types of cell division. Okay, and these two types, one is mitosis and another one is the meiosis. Okay, two type. This meiosis division, of course, the meiosis division occur only in the reproductive parts, and during the reproduction, this one is important. And mitosis is the common which takes place in the reproductive and the normal somatic body cells. So, in case of this mitosis cell division, this one takes place in animals. Mitosis takes place in diploid cells only. Cells only. Diploid cells means that the cells having 2n number of chromosome. They have 2n number of chromosome means two sets of chromosomes are there. But in plant, mitosis takes place in both haploid and diploid cells. Okay. So there is this is a very important difference in the plant and animal cells. Meiosis we will see later on. There is one term actually when these cells they divide. A cell is there, it may get mature, and this cell divide to produce two daughter cells. Okay, this one is called mother cell, which is dividing. Mother cell divide and give rise to two daughter cells. This daughter cell again, what will happen? This daughter cell again they will grow, mature, and then again they will divide. Next generation cell will form, and these next generation cells also they will grow, and then when they will reach to the maturity they will again divide. So there is division continuously. Cell divide, young one grow, mature, and divide. So every time these things happen, this one is given a term called cell cycle. So the process of the cell division, this goes on in a cyclic way, that cell it develop, it mature and divide. Again, grow, mature, and divide. This forms the cell cycle. So this is a sequence of event. Okay, this is there is a sequence of event. Here is you can see that what is cell cycle. Cell cycle, the sequence of events by which a cell duplicates its genome. Genome is that is the that they have genetic material. Okay, total genetic material in a cell. So it is all the genes in one set of chromosome. Genome is
Goodness. Thank God.
all all genes present in one set of chromosome. Chromosome is genes and chromosomes are very, very important things because they determine the character of the organism. So what happened that when the cell divide, there this genetic material also should divide, they should increase, and the daughter cell should get the equal amount of this genetic material, the chromosome. So what happened during this cell division, the genome they duplicate, means they make a copy. So there are it become two copies. So this is sequence of event by which a cell duplicates its genome, synthesizes other constituent of the cell. In the cell, some other things are there, different cell organelles are there, protoplasm that also should be increased. Okay, so they synthesize the constituent of cells, and eventually it will divide into two daughter cells. This process, that this sequence of event is called the cell cycle. So in a cell cycle, when the cell will originate by the cell division, they will first synthesize the material within the cell. So cell will grow in size. After that, they prepare themselves for the next division. And when they prepare themselves for the division, they have to first synthesize the genetic material. Okay, and also other cell component, and then finally it will divide into two daughter cells. Again the same thing will continue. So this is happen, and this cell cycle cell cycle has two things. The one is when there is a preparation for the division, the young one will grow, mature, and it will prepare it for the division. So this phase is called interphase. Okay? And the second step is the it is called M phase. M stand for Mitosis. Actually, this is the main things. This is the actual division. Mitosis is M phase represent actual cell division. Okay, and interphase is this the phase in between two successive division. That is the phase between two successive division. Okay, so this is interphase. So there are two phases, one M phase and another one is interphase. This here actually here actual division and here it is preparation and growth and preparation. Then within this also, this within the M phase and within the interphase also, there are sub phases. Okay, for the description, for the convenience of describing the process, this has given different name. So this interphase has interphase has been divided into three subphases. One is called G1. Then next is S and then G2. This G stand for gap. Okay. G is for gap. So gap 1, gap 2. And this one S stand for synthesis. Okay. Here in this gap 1, this is the phase immediately after completion of mitosis. That after completion of mitosis, that is the actual cell division, G1 start. And what happened in this phase, the cell synthesize the cell protoplasm, so it grow in volume. Here synthesize cell cytoplasm or cell protoplasm. Protoplasm and other organelles. Okay. It other organelles it produce. So it is actually here the young cells enlarge in size. That cell volume is increased. The material of the cell is synthesized and it attain the phase of maturity. Then this is After this, if the cell will divide, then the cell has to prepare for it, for the next division, and then it will start that it has to synthesize its genome, genetic material. Chromosome has to increase. Doubling of the chromosome or the genetic material is required. So in this S phase, the synthesis of DNA takes place. That is, here actually duplication, or we can say DNA replicate. It makes its replica, its copy. DNA replication takes place. Sometimes we call that the DNA duplication. Actually, this when the DNA duplicates, one thing we should remember that in a cell, like what we know that in a particular cell, there is a fixed number of chromosomes, is it? Cell of a particular species has a fixed number of chromosomes, fixed chromosome number. All right. Like that, a cell of a species, they have fixed amount of DNA. Fixed amount of DNA. And the content of the DNA is written with a symbol C, right? In a normal diploid cell, in diploid cells, the DNA content is 2C, right? DNA content is 2C, and haploid, in haploid just a minute, please. I have a phone call. Ah, hello, Malada. আমার খবরই নেই। এমনি ভালো আছি। তোমরা সব ভালো? হ্যাঁ, শিলংয়ে এখানেই আছি। আহ, আহ, মালেদা, আমি একটু পরে ফোন করি। একটা ক্লাস চলছে। না, একটা ক্লাসে আছি অ্যাকচুয়ালি। হ্যাঁ, আমি তো পরে ফোন করছি। আচ্ছা। Okay, I had one important call. Mm. So in a diploid cell, there is a content of DNA is 2C, and haploid cells, uh, like that, the haploid cells has DNA content only C, that is 1C, right? But when the when the cell is going to divide, it is preparing for the cell division, then they have to double their DNA. So this during this S phase, by the synthesis of DNA, the content of the DNA from 2C, it increased to 4C, right? It increased to 4C. But one thing we have to remember, that the chromosome number remain the same. Chromosome number is written by N. So chromosome number in the diploid was 2N, and during the preparation also the chromosome number remain the same 2N. Haploid cells have the chromosome number N, 1N. All right. So by the synthesis of the DNA, this DNA content become double and it is 4C. Okay. After the phase S phase, the synthesis, then the cell is in the G2 phase. G2 phase is this is a gap just before the start of mitosis. That is the actual division. During this process, of course, during this process, they synthesize the proteins. Here in the G2 phase, synthesize proteins for preparation of the cell division. They synthesize their cell organelles, okay, organelles, and also the cell volume, that also is increases. Cell size increases due to increase in the cell protoplasm, cell organelles, etc. Then after the G2 phase, the this cell will go to the M phase, okay, that divisional phase. Now one thing, that all the cells, okay, in our body, in case of animals or in plant, they all the cells do not divide repeatedly, okay. Many cells say, we know that there is a meristem in the root and in the stem, in the shoot tip and the root tip, there are meristem. In the meristem, the cells, they divide. Those from one cell, two cells are formed, one cell undergo maturation, it differentiate, and the other cell that again divide, again divide. So some cell and they forms, some of the cells they produce the permanent tissue, permanent cells. Those permanent cells they do not divide. In case of animals also, the cell initially they will divide, after that they will not divide, they will undergo maturation when the proper number is attained. Okay, so when this the cell after the division from the G1 phase, they may not go to the GS phase, because if the cell will not divide, it will not go to S phase. But what will happen, From the G1 phase, after the young cell has matured, young one will grow and mature, become adult. Okay, then, but they will not prepare for the next division. In that case, we call that stage is known as G0 phase. That is, here the cell exit the cell cycle, exit cell cycle, or it will exit the G1 phase. This is also called, that is the quiescent center, quiescent stage. Sorry, not center, quiescent stage. Hmm. This is G0 phase. Sometime, if it is required that the cell has to divide due to some cause, say sometime what happened, there is some injury, and in that place injuries to be healed. So new cells at cell should grow. During that time, this cells from the G0 phase can come back to the G1, and then S and G2, and thereafter it will divide. So all the cells which are mature and differentiated cell, we call that is the differentiated cells are in G0 stage. Okay, or the all the permanent tissue, they are in the G0 stage. Next, let us see this M phase. M phase, mitosis. Mitosis in the cell cycle, mitosis is uh, of a short period. Okay, whatever the time it takes that for completing a cell cycle, whatever time is taken, in that M phase is very short. Maximum, the interphase takes maximum time. Usually that interphase is for 95% time is occupied by interphase of total time. Okay, and for the mitosis it is what is left, that is nearly 5%. 5% time is for mitosis. So interphase is a long time, and in different species the time duration of the cell cycle is different. Say like human cells, no, human cells in culture take about 24 hours for completing a cell cycle. And out of this 24-hour, the cell division, mitosis is 1 hour, M phase. M phase is about 1 hour. So this one mitosis takes less time. Yeast cell, yeast cell, they take about 90 minutes. That is the cell cycle is very fast. It is 90 minutes. It's complete. 1.5 hours. Say bacteria, it takes about half an hour only, about 20 to 30 minutes. No, in bacteria, this is for bacteria. So different organism, different species, they take different time for completing the cell cycle. Okay. In M phase has different subphases. This M phase. Here it is. Again, in the M phase, when the cell is dividing, then there are two, the cell division, particularly this mitosis, we can describe in two headings. Okay. Two headings. So one is called the karyokinesis. And another one is cytokinesis. Karyokinesis is karyokinesis. This is the division of nucleus. Actually, caryon is the nucleus. So it is the karyokinesis is the division of nucleus, and this is the main part, main portion of the cell division. Here, that it is division of nucleus means the division of the chromosome. All right, division of chromosomes takes place, and then cytokinesis is the division of cytoplasm. This karyokinesis start first, of course, but later on, towards the end, karyokinesis and cytokinesis, they go side by side. And when cytokinesis takes place, then a one cell divided completely into two cells. So in this mitosis, we can, this particularly the karyokinesis, again we can describe in following heading. These are, one is called prophase, number two metaphase, number three anaphase, number four is telophase, right? This prophase, metaphase, anaphase, and telophase. This is at the end. And after that, we will describe this cytokinesis. Okay. So sometime this process of when the cell cycle is not asked, The mitotic cell division is described as prophase, metaphase, anaphase, telophase, and after that the cytokinesis is described. I will show you the cell cycle that see here from your book. You can see this cell cycle diagram. Can you see it? Hello?
Yes, sir.
Yes, sir.
You can see the page of your book, no? Here.
Yes, sir.
Hmm. So this diagram, this is here it is written diagrammatic view of a cell cycle. Say all these this has been shown by a like a graph, no, a circle, making a circle. It is showing. So this portion, this ring that which is containing the blue portion, this portion is the,
Interface.
the interface. All right, this is interface. Here I will show you this way. I can write. You can see in this diagram now. Now I can write it here. See here, this is 01 G1 phase. Okay, this is the beginning, and the process is going in this direction. Okay, here these two are the young cell which has been formed by the mitosis, previous division. Okay, and after that one cell has been taken and this cell is growing. This is for this it has been shown. So here is, this is the G1 phase, say how big it is. After that this is S phase. Here the synthesis of DNA, that is a DNA replication. Here this growth of the young cell, so the material, protein, etc. is synthesized, it's grow, and after that this is G2 phase. This is gap. All right, so this portion, that up to here, from here to here, it is interface. Okay, this is interface. Here you can see this around that arrow. Okay, showing a circle arrow. This one is G0 phase. It is showing G0, that if the cell will not divide further, then this will exit the cell cycle. That is, it will come out of the cell cycle and it will be in the G0 or quiescent phase. After the G2, here is the, this one is M phase. M phase is from here to here. This is a dark color line, right? If you see here in the entire circle, this M phase is, it is showing in the diagram, is only quarter of the entire phase. Is it? Entire cell cycle, it is only occupying only quarter portion. So in the M phase, this is prophase, metaphase, anaphase, telophase, and here showing cytokinesis. This, of course, cytokinesis is not separate. Cytokinesis, right from the anaphase and telophase, cytokinesis go side by side. So this is the, Here you can see the karyokinesis, cytokinesis. Okay, this has this showing interphase G1 phase, S phase of synthesis, G2 phase, some cell this about all these things I have told. No, G1 phase correspond to the interval between mitosis and initiation, that the 4C condition. In the next phase, you can say this is quiescent stage, that G0 when the cell is not dividing. After that is M phase. In the M phase, there is prophase, metaphase, anaphase, and telophase. This one is a very very important this division. What happened that this mitosis is also known as mm, mitosis is also known as equational division. It is equational division because in the mitosis, from one mother cell, two daughter cells are produced. Cells are produced, and these two daughter cells get the material equally. These two daughter cells, they are equal. Okay, they are getting from the mother, this mother cell, they are getting the equal content. That is, mother cells content are equally divided among the daughter cells. Whatever is the content is there in the mother cell, equally it is divided. And so the daughter cells are identical by quantity and by quality. Means they are getting the same quantity, and their quality also is similar, equal. So these daughter cells are equal in quantity and quality. Okay. So the DNA, that the chromosome, in terms of the genetic material particularly, in terms of chromosome, in terms of the DNA, both the daughter cell will have are identical. Identical. Okay, and that is why it is known as equational division. Say a mother cell, okay, mother cell having, this is the mother cell, it has say 16 chromosome, 16 number of chromosome. This will divide into two daughter cell. It is not that 50-50 will be given to them, because this daughter cell, this mother cell before division increase the chromosome number. So the particularly their uh, not the chromosome number, the DNA is increased, and this chromosome divide in such a way that their chromosome number also will be same. Both will have 16 chromosome, and the content of DNA also will be same. So what happen, these two daughter cell, they are identical to each other, as well as they are identical to their mother cell. Means daughter cells are identical to mother cell. So daughter cells also, they are identical to each other. Initially, this say DNA content remain 2C. This DNA content increase to 4C, and after that it is divided among the daughter cell. So it is 2C and 2C. Okay, 2C and 2C are produced. So they are identical. But I think today we cannot complete this cell division. If I start that, I cannot finish it. Only 2 3 minutes are there. In between, if you have any question, you can anywhere, any doubt. Do you have
No,
any?
sir.
No. So here from your book, you can go through what is quiescent stage, then this portion you should go through nicely. This portion, okay?
Okay, sir.
Ah, because where this what happened in the G1 phase, what happened in the S, what is 2C, what is 4C, a cell which is going to divide. Say based on this also, no, many questions are asked for the NEET exam, that is medical entrance exam. That what is the DNA content DNA content at the G1. DNA content in the G1, what will be the DNA content in G1? It will be 2C. But after the S phase, the DNA content become double. So in the S S phase and the G2 phase, the DNA content is 4C.
Right.
This 4C condition prevails for some time until and unless it is dividing into two daughter cells. So when the cell is dividing, that during the prophase, metaphase, okay, anaphase, uh, anaphase, the cell is still having 4C content of DNA starting from S phase. And after the telophase, again it come back to the 2C
Face.
stage. And if the cell is not going to divide, when the cell is in the quiescent stage, then it is in 2C stage, it will remain. Okay, after that, what is karyokinesis? Karyokinesis, what is cytokinesis, what is cell cycle, these things we have to yeast complete can progress through the cell cycle only about
3 minutes.
90 minutes cell cycle. This so I will today I will okay, stop it here, and next in the next class the